This is episode 23 of the Immunology Podcast, Dendritic Cells and the Immune System, Caetano Reis Sousa. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Roud. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. Today we have Dr. Caetano Reis Sousa from the Francis Crick Institute on the podcast to talk about his research in the molecular pathways that trigger dendritic cells into action. We've also got our usual roundup of recent highlights in immunology news coming up. But first,
Are you using dendritic cells for your research? With Immunocold Dendritic Cell Culture Kit from STEMCELL Technologies, you can generate immature as well as mature dendritic cells from human monocytes. Learn more at www.stemcell.com/DC Culture Kit.
excellent. Well, Brenda, how are you?
Hi, Jason. I'm doing very well. How
Good,
about
good.
yourself?
Okay. So, I guess let's see here. We're recording this right before Valentine's Day. So, do you have any plans for what's going to be coming up here?
Not really. Not a big Valentine fan here. I mean, many people are going on dinners on Monday, so I figure it makes sense to go on Tuesday or Wednesday. Would be a good time to reserve a spot. A lot of free places.
My wife and I have the same thought. We typically go the week after because, like, the food's all, not actually as good on the time of Valentine's Day. Sometimes you get a good deal on the price, but the restaurants are like overwhelmed, and like every restaurant in the world's buying the same steak or fish or something at the same time. We've never had good luck. Plus, I got promoted, and so we were going to go this weekend for the obligatory steak dinner. But then we're like, yeah, it's Valentine's Day, so like the week of the 20th, I think we'll do something.
Who needs Valentine's Day when you have promotion day?
Exactly. Well, it used to be for papers that we'd always go out, but now it's for other things.
Congratulations. When I grow up, Jason, I want to be like you.
Oh, thank you. You're close.
But there's something else coming up that is close to our hearts. AAI conference 2022. It's coming up in your hometown. I
Yes,
heard.
it's in Portland, Oregon, one of the best places in the world, like living in a postcard. So I highly recommend everyone go to AAI if you can. Some of us may be there. It depends on what various travel restrictions exist at the time. But check it out.
Just go ask for Jason. He's gonna buy you steak with his
Exactly.
salary bump.
Right. Yeah. We have a line now.
But yeah, so I'm not going. It's a bit far, but those interested, don't forget the registration closes on March 29th. And yeah, we're going to do something special for AAI. So just stay tuned. We'll have more information coming up soon.
Excellent. All right. Well, we got a bevy of papers to discuss.
Indeed.
I chose all things COVID just because, I feel it's been a few weeks since we did a COVID deep dive, and that's like a year in pandemic time.
I
Um,
know. It's been quiet. Not because it is really quiet, because here every COVID just exploded everywhere and all around. So let's see what the latest science brings us. Why don't you start?
all right, I may do mine too real quick, or we'll see if we want to go back and forth. You have a
Sure,
T cell
sure.
paper too, so here I'll talk about T cells and COVID, and then we can flip to your T cells.
Okay,
Perfect.
good
There we
enough.
go. All right. So this one is in Cell. It was accepted on 20th of January. It's still in the pre-proof stage. It is called T cell reactivity to the SARS-CoV-2 Omicron variant. Is preserved in most but not all individuals. First author is Vivek Dharandai, and last author is Gaurav D. Dehai. And it is a pretty good tour de force of immune effects from convalescent patients. So they took blood from people who had never been exposed to COVID, people who were infected with COVID, people who were vaccinated against COVID, and all combinations thereof. So infected and vaccinated, and so on. And then overlapped it with peptide pools from wild type and Omicron variant, as well as Delta spike variant, and tried to see what the cells would do, effector T cell responses and memory T cell responses, and effective boosters. So without getting super into the weeds, at a high level, it's an interferon gamma ELISA spot assay as their main measure. And plus, they did some CFSE proliferation studies. And long and short is that cellular immunity looks largely intact against Omicron post-vaccination or post-infection. In some cases, effector immunity is decreased in a subpopulation of people. So if you really parse things out, if you look at the average overall, it looks fine, but you can then see that there are some people that don't respond as well, particularly to the effector cell responses. And then they did epitope mapping and realized that, and then if you kind of look at those people, they have HLA subclasses that are more likely to bind to parts of the Omicron peptide, the spike peptide, that are changed between that and wild type. So they're more likely to lose the response from the HLA subtyping. But if memory cells are largely intact responses, booster cell boosting helps in all cases. So the number of people who don't respond go down. The magnitude of the response in people who respond less is increased. Generally speaking, everyone has an increased response with boosting. So it shows a lot of the real world data, but deep dives into it in kind of a neat way, and really does show that the immunological variation with HLA classing explains some of the fact that we're losing it. It's in those people with the right types of HLAs that then bind to the parts of the spike protein that are different between wild type and Omicron. Or Omicron.
Yeah.
I don't know which pronunciation we feel like using.
Then it would be the wrong type of HLA, I guess, for the purpose of protecting, of keeping your memory.
Yes. Yeah, exactly. So the different HLA, you won't keep your memory because it binds to the part that's changed. Fascinating.
Interesting.
Yeah. Why real life consequences of individual immunity plus population level effects of boosting. So there you go. What we're seeing in the
Fascinating.
world appears in the immune cells.
Fascinating. I mean, I find very comforting the fact that we, it feels a little bit like there's already been a bunch of papers showing that T cell immunity is holding the fort, and I find that comforting. And I think it's really cool that they actually found a reason for certain individuals to lose immunity due to their HLA types. And I think that's also very interesting, as you say, real life effects of HLA diversity. So thanks for bringing that nice paper to the table.
It's a good one. It's good to know what's going on. And again, it seems the vaccines seem to work really well to preserve immunity.
Okay, that's good. That's good for Pfizer's bottom line and Moderna's stock price.
And they had a bad stock readout recently. They're
I know. Yeah, I
anticipating weaker
heard.
demand.
It's an interesting. Yeah, it's an interesting time for biotech stock. Well, let's not get down that rabbit hole.
Yeah.
Do you want to continue with your next one, or should I talk about some real life consequences of T cell immunity?
You jump to T cells, and then I'll switch back to something else. I'll go to endothelial cells next.
Okay, let's just stick to the T cells for now. I want to share the results from a what was a kind of cornerstone in immunotherapy. Some of the first patients treated with CAR T cells back in 2010 in UPenn. You know that place, right? You've been there. Yeah, for our listeners who don't remember, yes. And there's been almost over 10 years since these first patients were treated, and some really cool analyses of the response of these complete responders has been characterized and published. So let's just talk a little bit about that. First, yay, 10 years since T cell therapy, CAR T cell therapy, big, I think it's a big thing, a big win. The paper is called Decade-Long Leukemia Remissions with Persistence of CD4 CAR T Cells. Gives away a little bit of the content. The first author is Josef Mellenhorst from where else? The lab of Carl June, a friend of the show, by the way. We have an interview with him a couple of shows ago. And basically, they evaluated the CAR T cells, the presence and the quality of the CAR T cells in patients that, as I mentioned, were treated over 10 years ago in 2010. These were two adult patients that were treated for chronic lymphocytic leukemia, CLL, which is actually not amongst the approved diseases for this therapy. So now it's only approved for B-cell, diffuse large B-cell lymphoma and acute lymphoblastic leukemia, ALL. I think that's probably the most known. And but these patients were treated in the context of a clinical trial, and they were infused with cells that had a CAR T cell, with the construct that is similar to the Kymriah, so would be the Kymriah CAR T cell that has a 4-1BB signaling domain. And they got fairly different doses. I think this is also very interesting. Although the first patient had doses of over 1 billion cells, 10 to the 9 cells, the second patient that also was a complete responder, and he's been cancer-free for almost 10 years, only received, and listen to this, 14 million cells, 14 million CAR T cells. And that was enough to handle his tumor. So if they look into the kind of history of these patients, both patients had pretty much undetectable B cells and lymphoma after 3 years. But the cells, they show that the cells stay there, the CAR T cells stay there for a really long time. Although they were kind of in the background, they were still a stable population of these CAR T cells many, many years after. So what they did is they did a kind of a very a wide CyTOF panel with 40 different markers to characterize the cells. And one thing that really comes up is that that fairly early on already, there started being a skewage towards a CD4 population. And by the end of these, so by the time of several years down the road, CD4 T cells became the vast majority of the CAR T cells. Almost 100% of the remaining CAR T cells so far are CD4 positive. They also had, in the case of the second patient with the lower dose, they observed at the beginning of the treatment a large population that was CD4, CD8 negative. And so, I thought that it was funny because they do a full RNA-seq with a site-seq, so markers, antibodies labeled with barcoding DNA to read, so to sequence this too. And they make a UMAP, they characterize the cluster just to find out that this CD4 negative, CD8 negative population was gamma delta T cells, which I would assume a gamma delta antibody would have shown right in their initial experiments. So I thought that was a little bit of an overkill, but still very interesting. So this patient also had a considerable gamma delta T cell population that was cytotoxic, and that also presumably was responsible to some extent of the tumor control. But by the year 7 and by the year now in 9, most of the CAR T cells, as I said, are CD4 cells characterized by a high expression of Ki67 and other markers related to T cell activation, including CD38, HLA-DR. Interestingly enough, TOX, EOMs also, and CD27 and CCR7, which are usually were associated with kind of this long lived memory T cells. So it was, I think, maybe related to their high longevity. Well, they also looked into they did analysis looking into places where the transgenes integrated. They saw that at some points they did disrupt the expression of certain genes, but it wasn't like the TET2 story that also came from the Carl June lab, that they found that it's very specific knockout of genes became dominant in the T cell populations. Although in both cases the populations are very oligoclonal, so a very few clones that dominated the CAR T cells by this time. So so I think, and just as a final thing that I thought was very cool, is that these CD4 T cells, they're activated, they are highly proliferative, and they're also cytotoxic. They're expressing granzyme K and granzyme A, and also perforin, which is not unlike maybe granzyme B, that I think is more commonly evaluated for cytotoxicity. And this reminds me of a paper that came out last year, in which they're also looking into CD4 cytotoxic cells, and I think it was in context of bladder cancer, and they also found this kind of curious combination of cytotoxic markers. So I thought that in general, the paper is very small, so anybody interested in kind of this very breakthrough science and just to understand a very long story, it's a nice paper to read. And I just thought, well, it was really nice to be reminded that it's been 10 years since these patients were given a second chance.
I think that's really important context there of just the time scale we're on. But also, you make a good point of you could have just run an antibody. Remember, to get a high impact paper, you have to have single cell RNA-seq now.
I guess.
So
I
you just
guess.
do it, right? Just throw it in the beginning and you call it a day.
Yeah. Who needs antibody development when you can just make a UMAP and just find your clusters there, right? I feel so old school.
Exactly. All right. Well, out with the, I guess is CAR T cell therapy old now at 10 years now. Out with the adolescent, and in with the super new still, back to COVID with endothelial cells. So I guess I'll give the title and then I'll give some context and I'll dive in. So this is extracellular vimentin is an attachment factor that facilitates SARS-CoV-2 entry into endothelial cells. It's in PNAS. First author is Razi Amre, and last author is Nader Rahimi, or Nadir. And it was published February 8th of this year. So before we go into further, the reason I found this interesting and I went after it was because we have known for some time that COVID isn't just a respiratory disease, it's also a vascular disease. And that is been one of its key problems, right? So it infects the vasculature of your lungs. And so regular ventilation didn't work very well. That's how we lost so many patients early on. We were ventilating like pneumonia. Doesn't help. Doesn't help to put more oxygen in the lungs that were just fine. You're actually damaging them with ventilators when the issue is that you have a perfusion mismatch and the blood can't get there to exchange oxygen. You have heart attacks, strokes with COVID. You even see it with the vaccines that the biggest side effect is some form of blood clot. Sometimes it's the most concerning, or heart inflammation, right? So there's which is not endothelial, but you're still talking, circulatory system. But like why it affects the endothelium, what's going on? Because ACE2, which is one of the main receptors, isn't that highly expressed on lung. It's there on lung, but more upper respiratory than lower respiratory. And it's there on endothelial cells, but it's not like, oh, the most important receptor ever. It's important there, but not the only thing. So it's not like it is the highest expressed there. Of course, it goes to the endothelial cells. It's like, yeah, it's there, but why is this hitting the endothelium so much? And so this paper helps answer that, which shows that extracellular vimentin, which is on endothelial cells, expedites SARS-CoV-2 entry via the spike protein into cells. So vimentin binds to the spike protein, and then through a co-interaction with the ACE2 receptor, helps COVID or SARS-CoV-2 get into cells. And there's your answer. So the paper goes through, it's mostly cell work in 293 cells, some lung cancer cell lines. They use an antibody that blocked vimentin interaction with COVID that was known to affect that binds the spike protein in a different spot of the receptor binding domain area, but a different spot than where ACE2 is known to specifically bind, and showed that instead of blocking the same site, so it doesn't block the ACE2 binding section of the receptor binding protein, it blocks another site. And vimentin really does serve as a cofactor. So they take proteins, they put it on, and they take cells that overexpress both or one, and then use either COVID binding receptor binding domain to show, ligand interaction, or pseudovirus or virus to show infection. And through, all the permutations really demonstrate that this vimentin is responsible for increased uptake into cells. And they map it out. They map out and then I guess the key from the biochemistry again is then they use neutralizing antibodies and show that antibody, which is already known to bind to a non ACE2 section of the receptor binding domain, knocks out the ability of vimentin to do its thing. So it's showing that it's a separate cofactor. So very interesting. Suggests that there's a whole bunch of more biology going on with viral entry. They do all types, they use bait and trap proteins to find vimentin to begin with. They use a bait approach to see what will bind to the receptor binding domain besides ACE2. Pull it down, find out that it's vimentin by mass spec, put it into cells, go through the whole thing. So really solid viral, immunological biochemistry, which is my happy land. Very well done. Like what it's showing, provides some really important clinical information, suggests that you could have antivirals that not just block ACE2, which has consequences, maybe block the other parts of the spike protein. So it's starting to get into some interesting biology here, for me at least.
Very interesting. Thanks for sharing. I I like how there's more attention now paid also to the whole systemic disease that COVID really is, and I think it's only now that general public is realizing that, because it's been a mistake, I think, to always frame COVID as a respiratory disease, because yeah, as we see, the most dangerous, I think, effects pathology is actually what happens in your vasculature and as such, and the kind of maybe a little bit more hidden, and people don't realize that. It's very interesting to see that we can identify factors that explain this tropism that maybe is not so self-evident.
More uncovered every day.
Yeah, yeah, it is. I had to say, lately, it's so hard to keep up. There's so much being published, and feels like everything is so important. Everything kind of solves a little piece of the puzzle, and increases and enhances our understanding of the disease, that I have to say I feel a little bit outdated, especially when I was on holidays. I didn't read that much, so I feel like a month came in, Omicron came in, and then so many things happened, and I'm just not up to date enough. Oh wait, but I'm boosted, so I guess that
You're good for
that will get
now.
my T cells will take care of me, hopefully.
Remember, T cells
Um,
get the job done, right?
yeah, for sure, for sure. I mean, so far I haven't gotten it, and I've been surrounded. I've been completely surrounded by it.
Yeah.
So
Well, I got
maybe
Omicron, but it was not that bad.
yeah, that's what I hear. So still, not necessary to get it. I don't think it's an experience I necessarily want to have. Talking about experiences that I wish I had, which is making this amazing engineered T cells that I'm going to talk about now. This is a work that comes from a lab that I admire very much, the lab of Alexander Marson at UCSF. They are complete rock stars when it comes to engineering T cells with CRISPR and non-viral methods of modifying T cells. And they have been doing a lot of really cool stuff lately, a lot of screens, CRISPR screens, looking into T cell function. And I want to talk about one of those today. First, authors Ralph Schmidt and Zachary Steinhardt, published in Science, I think, well done. And it's called CRISPR Activation and Interference Screens Decode Stimulation Responses in Primary Human T Cells. And I think that this is mostly a technology paper, so to say. They're really showing how far you can push the tools, and I think that is very admirable. They do make some nice points about the kind of research that you can do with these tools. So let's just dive in. Uh, Basically, what they are showing is the results of what I assume are years of optimization and study of a lentiviral. So it's a lentiviral mediated um, CRISPR screens for primary T cells, which is not that easy. They have been able to have very high transduction efficiency, so to really have a functioning protocol for screening really large libraries. And what they basically, kind of a quick overview, what they do, they generate these libraries for testing CRISPR activation and CRISPR interference uh, tools. So how does this go? They have modified Cas9s, which are this non-endonuclease capable, so this dead Cas9s, and they either couple them to the VP64 transcription activator into the C terminus of this dead Cas9, or they fused it to a Kerpel associated box, so a crab repressor, that to induce repression of a particular gene. And so they started their screen with a screen targeting over 18,000 protein coding genes with over 100,000 single guide RNAs. So huge library, very, very impressive. And what they do is they look into the production of two particular cytokines, either IL-2 for CD4 cells or interferon gamma for CD8s. And using this, so they sort cells into the highest and the lowest producing for each. So after the cells being transduced with this library, and they use this to identify regulators of the production of either of the cytokines. And the truth is that by now we have most of the pathways of that mediate of that influence the production of IL-2 and interferon gamma pretty much figured out. But they show that by doing this CRISPR activation screen, and they also kind of do the opposite. They do a CRISPR interference screen. They can basically reconstruct the major elements that regulate the expression of either of the cytokines. And I think they have a really nice example of how from this data they can re-identify a majority of elements that influence, for example, the NF-kappa beta pathway that regulates interferon gamma production. And they can find this all from their screen. And I thought we already know a lot about this pathway, so there's not a lot of new information. But if you're looking for a different pathway and you want to identify missing links, maybe this is the way to go. They also look further. They validate this by using arrayed experiments, where they assess also they choose some of the most interesting targets, and they look into the production of other cytokines, and they just use this to generate this really complex web network of factors that, again, a lot of this is already known, but this is something that they do based on only these screens. And what I think that they conclude on, which is very nice as a tool, they define what they call Perturb-seq, which is a platform that couples the pooled, so like CRISPR-A perturbation, so this library of CRISPR activators, and they pull them with barcoded single-site RNA-seq readouts. So what they can do is they can just do all in ones. They transduce the cells with this activating library, and then they can just with the same RNA-seq, a single-cell RNA-seq, they can identify which is the guide RNase that is targeting in this cell, and then have all of the transcriptome data that allows them to do all clustering and characterize the cells. So it would be instead of only evaluating one particular known readout, you can just do the whole transcriptome and see how each of the perturbations affects the whole thing. And so they can also characterize cytokine activation scores or things like that. And then you can just really in one go look at the effects throughout the whole transcriptome. I thought it was really cool paper. Really interesting to see what kind of things you can do with this technology. And I'm looking forward to see what they're going to apply it with and what kind of questions people can answer with this platform.
So do they have any teases of things they've seen with this one in this paper besides here's the platform, like, ooh, we found this pathway that was unknown or only hinted at, or?
I think they do. So if they go through the paper, they do mention some things. So for example, they find a particular gene, IKZF3, which, for example, reduces the production of interferon without reducing the overall activation score. Because in many of the cases, what you get is some element that is associated with T-cell activation in general, and therefore it also affects interferon gamma expression and NF-kappa beta activation. But for example, by using this, they identify a particular element that affects interferon gamma expression without messing with the activation score of the cell.
Interesting. All right. Well, we'll have to see what comes next.
Yeah, for sure.
Well, for us, what comes next is a great interview about dendritic cells, which we haven't gotten to talk about a lot.
Yeah.
We're going to be speaking with Dr. Caetano Reis Sousa at the Francis Crick Institute. But before we get to that, do you want to differentiate human pluripotent stem cells into monocytes? Check out the STEM-Diff monocyte kit from STEMCELL Technologies. It generates millions of monocytes that are ready for downstream assays or for further development into macrophages or dendritic cells. Learn more at www.stemcell.com/monocyte-kit.
Today we're joined by Caetano Reis Sousa. He's a senior group leader of the Immunology Laboratory at the Francis Crick Institute in the UK. He and his lab have made really seminal contributions to our understanding of dendritic cells, particularly conventional dendritic cells and CDC1s. Really exciting work came from his lab in this field. He's joining us today from London. Thank you so much for talking to us.
It's my pleasure. Thank you for having me.
Thank you for being here.
Well, I do like to think that all immune cells do deserve their spotlight, and I feel very what's the word, very aware that dendritic cells have not had enough attention in our interviews. So I'm really happy to get to talk to you today. So I just want to hop in right away. And maybe you can start with sharing with our listeners a little bit about what has been your understanding, or what is our current understanding of dendritic cells, particularly conventional dendritic cells, and their development and their function, especially when it comes to cross presentations and those subsets that are most recently been characterized, for example, as being important for cancer or other initially less understood functions?
Well, that's a very big question. So let me try and break it down into smaller bits, perhaps. I think perhaps the first point to make is that there are many different things, many different cell types that are called dendritic cells, and there's no universal definition of dendritic cells. And so some people will perhaps disagree with what I'm about to say, but I think that it is perhaps easiest when talking about dendritic cells to consider the ones that were originally defined as such by Ralph Steinman nearly 50 years ago. And despite all of the diversity in types of cells that have since been called dendritic cells, we now think that the ones that Ralph was referring to are probably most likely the conventional dendritic cells, which is why this term conventional has really come into being, classical or conventional cells. And if you think about those, I think you would exclude anything that is a monocyte-derived cell, including things that have been called monocyte-derived dendritic cells. You'd probably exclude also the plasmacytoid dendritic cells, which we have started to refer to as plasmacytoid cells instead of plasmacytoid dendritic cells. And then you'll be left effectively with two basic flavors of these conventional dendritic cells, and those are the CDC1 that you've alluded to and the CDC2. Now, it is true that all of these, including monocyte-derived cells, including plasmacytoid cells, play key roles in the immune system. But unless we have some way of thinking about them and referring to them unambiguously, I think we tend to muddy the waters. So going back to your question about functions of dendritic cells, the other thing that I think we have to consider is how many functions might be shared amongst cells, even amongst different flavors of cells. And the other one is whether there are certain functions that might be unique to certain subtypes of cells. So the CDC1 have attracted a lot of attention because they seem to perform some non-redundant functions in cancer immunity. which is not to say that they can't do additional things that DC2s can also do. And it is also not to say that DC2s can't also play a role in cancer immunity.
So, kind of going into some of your work in relation to this, and by the way, going through your work, there was a lot. Yeah, I think you had talked about this. I'm going to probably botch the pronunciation of this. Is gelsolin? Did I get that right? Or gelsolin?
Yes, gelsolin, correct.
Okay,
Yeah.
gelsolin. All right, good. So I got there. So I think we're going to range quite a bit across some of what you've studied, but could you talk a little bit about this? This was a really interesting paper where you found a pretty good single molecule, as in a single peptide that had an important effect that is druggable, which is often like the hardest thing to find.
So there's quite a bit of background to this, Jason, and it goes back a little bit to Brenda's question about DC1s in cross-presentation. So as we all know, cross-presentation is the ability to display exogenous antigens as peptides on MHC class 1, which in many cases requires those antigens to get access to the cytosol of cells, which seemingly violates the rules of cell biology, where things from the outside end up in endosomes, but not in the cytosol. So quite a few years ago now, we found a receptor that appeared to be very restricted to DC1s, although since then we've also found it to be expressed in dendritic cell precursors in the mouse, for example, but very highly restricted to DC1s in both humans and mice. And what this receptor appears to do is to actually selectively bind to dead cell remnants that a DC1 might encounter. It does so by binding to exposed F-actin on those remnants. And when it encounters those F-actin remnants on a corpse or that's been internalized by a DC1, and this can happen now within a phagosome, it actually can signal to favor the rupture of that phagosome and release of those contents into the cytosol, thereby allowing them to enter the class 1 presentation pathway. So this will be a fairly simple mechanism for cross-presentation. But the key point is that this is not dependent necessarily on just unique cell biology of DC1s, which has been previously argued, but dependent on a particular receptor that they express. And we can put that receptor on other cells and make them behave exactly the same way. So perhaps not surprisingly, the properties of the DC1s are to some extent going to be dictated by the types of receptors that they express. And this is one example. Now, it turns out that the ability of this receptor to bind to F-actin is actively antagonized by another protein called gelsolin. And in fact, gelsolin can be secreted by all cells, including cancer cells, using a alternatively spliced form where one of the exons contains a signal sequence. And this secreted gelsolin, as it's called, turns out to potently block the binding of DNGR1 to F-actin. And consequently, what we found is that it's also able to block this cross-presentation by DC1s, including in contexts where we're studying anti-cancer immunity. So it acts as a sort of dampener of this activity of DC1s in cross-presentation of dead cell-associated antigens, and in so doing, it also can dampen anti-cancer immunity. So yes, as you point out, that is potentially a druggable target if we can find a way of blocking the ability of secreted gelsolin to inhibit the NGR1 mediated recognition of F-actin. Then we could potentially augment this cross-presentation and augment anti-cancer immunity. We haven't done that pharmacologically, but we have shown that genetically, that by deleting secreted gelsolin in mice, we can actually potentiate anti-cancer immunity in those mice. So it is potentially an interesting way of thinking about boosting anti-cancer immunity by boosting dendritic cell-mediated cross-presentation.
I also thought it was very interesting the fact that you find a similar receptor as kind of far off as in Drosophila flies, you can find also receptors that are binding to proteins that indicate cellular damage, tissue injury. I thought that was really cool how you could find this homology way out in the animal kingdom.
That's right. So I mean, just to clarify, there is no actual homologue or ortholog of DNGR1 itself in Drosophila. But what we have found is that Drosophila nonetheless possesses the capacity to respond to the extracellular presence of cytoskeletal elements, not in fact actin, but an actin-associated protein called alpha-actinin. So the principle, as you point out, is indeed conserved throughout evolution, and I guess it makes sense, right? Ever since the evolution of metazoan organisms, we have had a need to recognize tissue damage, to put in place tissue repair, and to effectively ensure that organs are restored and organ function is restored. So the fact that this exposure of cytoskeletal elements seems to serve as a sign of damage as far back as invertebrates, and then it's co-opted also by the immune system of vertebrates, does indeed suggest that this is a common theme, and I think does lead us to think about ways in which we can think about cytoskeletal exposure in the context of various diseases.
And one more question on this topic. Another very key question that your lab has addressed related to CDC1s is to understand better the mechanism of cross-presentation. Because I remember, especially like there's so many, I have seen so many slides in which they say, yes, there is cross-presentation and there's a big kind of question mark in the between the entry of the antigen and the presentation. Nobody wasn't really clear how this crossing was happening. But your recent research did give us some really nice insights, especially in the case of DNGR1 mediated cross-presentation. Would you like to maybe share that too?
That's right. I mean, I have to say that personally, this gives me a huge degree of satisfaction, because my first project as a postdoctoral fellow in Ron Germain's lab was to study cross-presentation at that time in macrophages. And actually, at that time, we came up with a very simple hypothesis that we called the indigestion model. We said, there are a number of proposals that have been made for cross-presentation, including that perhaps particular channels in phagosomes are specialized to transport polypeptides that are generated from cargo that's internalized. And Ron and I actually said, well, that may well be the case, but in fact, another very simple hypothesis is perhaps some of these phagosomes just burst, especially if there's overloading of the endocytic pathway, which is why we call it indigestion. And I think we even wrote about this rupture as a possible ulcer. And then many, many years later, we are studying this receptor, DNGR1. And in fact, it turns out to be a receptor that can control cross-presentation. And that's independently of cargo uptake. And I think actually a very neat experiment that we were able to do that sort of, I think, proves that point is if we feed DC1s that express the NGR1 with latex beads that contain ligand or not for the NGR1, we can find that the beads that contain, the cells that internalize beads containing ligand cross-present better. But of course, that could just be that they take up the beads better. So actually what we did is that we sorted the cells that had taken up precisely one bead, and we could find those cells because the beads are fluorescent. So we could actually take cells that took up precisely one bead. So all the cells had exactly the same content of antigen, which was bound to the latex beads. But one group additionally had the ligand for DNGR1 and the other one didn't. And the ones that had the ligand for DNGR1 actually cross-presented better. So that told us that whatever was happening was post-uptake. And after quite a bit of detective work, we found that in fact DNGR1 was signaling to promote this indigestion and this rupturing of phagocytes. Now, it's not an all or none phenomenon. It is basically causing localized membrane instability, we believe through lipid oxidation by engaging NADPH oxidase. And that increased oxidation of the lipids in the phagosome just raises the probability that the phagosome will rupture. So not every single one phagosome will rupture, but some of them will. And at that point, your material is in the cytosol and can access the proteasome. And we believe that this is actually a fairly simple mechanism that might be applicable to other cases, not just the NGR1. And I think it also tells us something quite important, which I don't think we had fully appreciated, because people had pointed out for a while that CDC1s are very good at cross-presentation compared to other cell types. And I think that was interpreted to mean that they have a sort of constitutive ability. What I think these experiments show us is that some of this is inducible. In other words, it depends on a receptor actively signaling in response to its ligand. And it's because the receptor tends to be restricted to DC1s that perhaps they come up tops in a lot of assays of cross-presentation.
All right, well, I'm going to shift gears a little bit because you've had a very busy 2021, and I can't just talk about your cell paper. We also have to talk about your science paper. So I was really interested in the dicer protein that you put out with the isoform.
Yes.
As we know, RNAi is really important for viral immunity in most critters, but hasn't really been established in humans. And we have interferons, but why don't we have RNAi? Because if you do it exogenously, it seems to work just fine, right? You can do it, you can do it with cells, but it's not like a thing. But apparently, it is a thing. And so, could you describe from your very interesting science paper how you established it was indeed a capital T thing for this alternative splice dicer?
Well, Jason, I think you just gave the whole background to it, which is precisely the way that we were thinking. As you point out, RNA interference is the major mechanism utilized by invertebrates and plants to defend themselves from virus infection. And we have all of that machinery there to execute RNA interference. We have the Argonaut complex, and one of the Argonaut proteins, Argo2, is capable of endonucleolytic slicing. So everything is there. Why is it not being used? And so a number of researchers had actually proposed that perhaps RNAi is active also in mammals. That became very controversial. It actually became a very heated discussion in the field with people for and against. And I've always been interested in responses in particular to RNA viruses from our historical work, where we found that TLR7 was the receptor for RNA in endosomes. And then we later on found that RIG-I was able to respond to 5' triphosphorylated RNA and diphosphorylated RNA. So we had always had an interest in how the innate immune system responds to RNA viruses. And that, by the way, was quite independent of our work on dendritic cells. So someone, Pierre Maillard, came to my lab quite a few years ago with the notion that he wanted to explore RNA interference in mammals, which he had been doing already previously. And he and another colleague, Anna Martha van der Veen, provided some evidence that the system was there, present in mammals, but masked by the interferon response, which is so powerful. And that's where we had left it until another individual, Enzo Poirier, came to the lab more recently and wanted to look at this further. And our findings up to that point did suggest that dicer-mediated dicing of the RNA was really the bottleneck. That's where the inefficiency came into it. And it's interesting that organisms such as Drosophila actually have two dicers. They have a dicer that is involved in the microRNA pathway, much like our human dicer. But then they have a second dicer, dicer 2, that is specifically co-opted for antiviral RNA interference. But we only have one gene encoding dicer, so not two. And so Enzo and I had discussed the possibility that there might be two gene products from one gene. And sure enough, he found that there was an alternatively spliced form of dicer that effectively was more efficient than canonical dicer at chopping up viral RNA or viral or double-stranded RNA analogs of viral RNA, and was able to effectively more efficiently carry out the antiviral RNAi. Now, what was really interesting is that it turns out that this form of dicer, which we've called AVID for antiviral dicer, this form of dicer seems to be preferentially expressed in stem cells, including tissue stem cells. In fact, we cloned it originally out of neural stem cells. And one reason for that may be that stem cells don't rely as much on the interferon response. The interferon response tends to be cytostatic and cytotoxic, and perhaps you don't want to lose all of your stem cells if they engage that response. So they seem to be hypo-responsive to the interferons. And this may be a sort of primordial antiviral mechanism that they fall back on, and they use it preferentially. That, of course, begs the question, why don't just all cells use both? Why not have interferon response and antiviral RNAi? Why should they be antagonistic? Why should interferons suppress the antiviral RNAi response? And we don't have a good answer for that, except to say, and we had already proposed this a few years ago, except to say that the interferon response relies on detection of these long base paired RNAs, including double-stranded RNA, which induce all of these antiviral effector proteins. But then the antiviral effector proteins, for example, PKR or RNase L or others, still require the activation by the double-stranded RNA in order to perform their function. If antiviral RNAi was involved in chopping up all of the base pair RNA derived from the virus, then perhaps these other proteins could not then become activated after being induced by interferons, and therefore they couldn't exert their effector function.
So it looks like you've essentially found pretty solid evidence as it happens. Is there any follow-up that your guys are doing more in humans to really show how well this works? I know, like, you can't obviously go give them RNAis and see what happens, but I don't know if there's a way to look at this in patients in some form, like SNPs that affect the ability of this protein to function, infection rates, or God knows what.
Yes, I mean, we haven't actually been able to do very much with human patients or even some of the sort of genetic type of mining that you bring up. We have used, of course, human cells as well as mouse cells. Our strategy right now is to try and make a mouse model of this and see if we can actually make a mouse model in which we get rid of AVID and then look for loss of function in a number of different diseases, viral diseases. I should point out that Enzo Poirier, who carried out that work, has now moved to Paris, where he's continuing some of those studies. And he may well think a bit more deeply about how we could eventually move that closer to humans and potentially exploit it in some form of therapy.
All right. So the next one to dive in, I'm just going to go to your other Science Immunology paper for 21, and I guess I think maybe we'll have covered most people's lifetime of work, but a year for you. So this paper you guys look at talks about how to sustain immunity to a pathogen, in this case influenza, because why not use a relevant one? You have to have the dendritic cells get there and do their magic. So can you explain top line, I guess, why this, I think why it's important is clear. You want to sustain immunity. But what was, and this is where I, as a non-dendritic cell expert, what makes this extra surprising, right? Because when I think about, like, of course you want all the immune cells in the places they need to be to do the things they do. So what was like the big aha moment here for this one that like really, I think, helped solidify our understanding of dendritic cells?
Yeah, so I mean, I think actually in retrospect, as you point out, you sort of go, yeah, of course, it has to be like that. But what I don't think had been articulated before was that in fact this is needed. So every textbook of immunology that I know of will show that dendritic cells, and will state dendritic cells are sentinel cells. They're present in tissues and they are mobilized in response, for example, to infection, where they effectively transport pathogen antigens and pathogen signals into the draining lymph nodes, where they can then prime T cells. I haven't really seen anywhere in those textbooks the next sentence that says, but by the way, there's not enough dendritic cells in tissues to do this. And therefore, at the point where you are infected, you need to call for backup and ensure that you get more dendritic cells to make sure that you've got enough sampling capacity for all of these pathogen antigens and all of these signals to then sustain an immune response. And by the way, if you interrupt this backup, you're in a bit of trouble, because you will get an immune response, but it won't be quite of the magnitude that you need, and it won't quite generate the memory that you need. So that was our aha moment, as you put it, which is the realization that, of course, you can't have a tissue chock-a-block full of dendritic cells. You need to have just enough to get things started. And what you then need to have is a system that cleverly calls for backup, but it's very localized. And that backup, by the way, comes all the way from the bone marrow, because, of course, that's where the dendritic cell precursors develop. And that's the place from which they have to leave to then colonize the tissues. And in the steady state, in homeostasis, this is a freewheeling cycle. It's happening all the time at some basal level. But I think what we found is what we've called emergency DC-poiesis, which is that in situations of demand, you can greatly accelerate this mobilization of these precursors, so that you've got increased numbers of them being released into the blood. And what we found in that paper is that, quite remarkably, they are directed specifically to the sites where they are needed. So in the case of influenza infection, you actually direct them specifically to the lung. So you've got these precursors. They don't go to the spleen. They don't go to other places. They go to the lung, where they increase in numbers. But even more remarkably, they go very specifically to the foci where the infection is happening. So you find them now adjacent to the sites where you see viral antigen, where there's viral replication. And this process, incidentally, is controlled by CCR2, a chemokine receptor that we have generally associated with monocyte mobilization, not with dendritic cell mobilization. And again, just to go back to something I mentioned at the beginning of this podcast, I'm talking about conventional dendritic cells, not about monocyte-derived cells that people have sometimes called dendritic cells. So these are the canonical, bona fide, conventional dendritic cells that are being called to those sites where they are needed. And if you will, this makes a lot of sense. If you have a whole lung, even if there's quite a few dendritic cells in that lung, it's a large space. And so if you've got infection happening in some alveoli, you can't really get dendritic cells from other alveoli far away to traverse the tissue and get to where they need it. The only way that you're going to get more at that site is if you get them from the blood, because the blood, of course, is irrigating every part of the lung. So that's precisely what you do. And we're very interested right now in understanding this process of emergency leukapheresis and what signals control it, because you could see how you could harness that potentially in vaccination. You could, if indeed you need more dendritic cells than what's there to actually sustain a strong immune response, then if you figure out what are the right signals to deliver to the particular tissue where you're depositing your antigen, then you might actually increase the efficiency of your vaccine. And just as a final point, we found that in the case of flu infection, it was CCR2 that was important, but actually that is not universally the case for every challenge. So for example, in another model of parasite infection, which is also affecting the lung with Nipastrongis brasiliensis, which we also showed in that paper, you still have this recruitment of dendritic cells as acute backup, but in that case, it's not CCR2 dependent, and we still don't know what it depends on.
That is really fascinating. And well, as I say, if you put it that way, it makes sense that they'll go there. But I guess that you don't know until you don't see it and until you don't prove it. So very fascinating. I think we could keep talking about your research for a really long time. There's always there's a lot to talk about, but I think we're gonna call it a scientific day for now. But before you go, we would like to ask you a tangent question. And we would like to ask you, what is the biggest misconception about science that you would like to resolve?
I don't know if it is the biggest, but it's one that I feel strongly about, which is the concept of science and arts and humanities as separate human activities. And you often find the so-called intelligentsia of various countries sort of proudly declaring, oh, I know nothing about science. But if I, as a scientist, say, oh, who was Beethoven, or who was Picasso, they would think that I'm an ignoramus. And I think this notion that we have these different human activities is something that needs to go. I mean, what I think is fascinating about science is that it's really about our humanity, is what effectively distinguishes us from animals is our curiosity about the world that surrounds us. And that curiosity is the same that drives the arts and the humanities. So from the beginning of time, humans depicted the world around them. That's the origins of art. The paintings in caves are effectively a depiction of the world, and with that depiction of the world comes the wish to understand that world, and that's the basis of science. So for me, science is really just a continuum in human activity, and I think that there's this big misconception that we should compartmentalize these things, and I think there's quite a few people now who actually are looking at these interfaces between art, science, literature, music, and I think that we're all the richer for that.
That's very, very. I agree with that very much. No, you should plan the comeback of the Renaissance man or woman. But I think also this idea of separating the hard sciences from the arts, it's kind of recent, because if you looked at all the scientists from the 15th, 16th century, they did it all, and they didn't see a reason not to go into all the fields.
Absolutely. And Leonardo da Vinci was an amazing painter, inventor, anatomist, scope. I mean, that really is, I think, how we should be thinking about science as part of this continuum.
Although I think it gets difficult because, back then a scientific discovery, like how many figures in panels are in a paper now? You really are. You're an immunologist and you're a molecular biologist and you're a cell biologist and you're a chemist, and like to do one thing, it's so expanded. But yeah, no, I agree that we've kind of, it's not as narrow as people think science is, but simultaneously, I think to just get something out now requires so much. You're almost a Renaissance human of science to be able to do anything.
Indeed.
Thank you so much for joining us, Caetano, today. And we enjoyed the conversation very much.
And thank you as well. It's been very interesting. And I'm very fascinated about the dicer stuff. I think that's really cool to find, I have to say.
Well, thank you very much for having me. And it's been a great pleasure.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all of the interview and Roundup papers. You can also reach out to us on Twitter at @immunopodcast or via email at info@immunologypodcast.com with feedback or to suggest guests. See you next time.